want to just start with some acknowledgements. This is my group. It's been a fantastic pleasure working with everyone here. Yeah, there's a lot of work that I'm not going to be able to tell you about, which makes me sad, but it's been a pleasure working with everyone. And the work I'm going to be telling you about today, the majority of it was driven by my first to graduate student, Sam Bryant, who's here. And then at the end, I'm going to tell you a little bit about some ongoing work, not yet published, from Jose Betancourt. And with that, I'll get into it. Okay, so you're about to see a movie of some swimming bacteria. Let me just play this. And I'll tell you just a little bit about it in a second. But first of all, there's a limit that you can say about the motion of these critters really without knowing anything else, even the scale bar, and that's that they have to be going slower than the speed of light. So that's a bound that you can always say, but it's not a useful bound. So these are about a micron long. The flagellum in the back is maybe 10 times longer than that. And they go about 13 orders of magnitude slower than the speed of light. But if you do know something about the medium they're in, that it's high viscosity, they're small, so they're at low Reynolds number, then you can actually learn quite a bit about the strategies they use, why it's efficient subject to the constraints that they live in. And there's this very nice paper, Life at Low Reynolds Number, that explores that. This isn't what I'm going to be talking about, but what I want you to take from this is that not all bounds matter. But if you think carefully about the constraints that do matter, you can make progress in understanding how these systems work. Okay, so this talk is about processing information and the constraints that act on that. And I want to start by saying that there is a fundamental bound on the energy cost of computation, but it's very small, and it's hard to apply. That's not what this talk is about. And it turns out that copying information is free. You can do that in a reversible way. You can duplicate information in a thermodynamically reversible way. And in papers about this, you see figures like this, where you very slowly and adiabatically change the depth of two wells. But if you then want to erase a bit of information, that costs kT log 2. So that's about 10 to the minus 20 joules in the cellular environment. And this places a bound on the cost of processing information if you do it in the right way. But the measured costs are very much not of order kBT. And these are some nice papers from Laughlin and collaborators from the late 90s. And what they did is they took a photodetector cell, so this is a light-sensitive neuron, and they played it a little flickering movie. And then they could measure the voltage across it and see how much information the cell is getting about the movie. And at the same time, they can measure the energy, the oxygen consumption of the cell. And they can subtract the amount it uses when it's not seeing a movie. And they can infer how much energy it is spending to transmit a bit of information from one cell to the next. And what they find is that this is something like 10 to the 6 to 10 to the 8 kT, depending on the neuron, to transmit a bit. And they have a very nice practical accounting for this. So the main costs are actually recycling and packaging neurotransmitter into vesicles. So the way they send this information eventually is to release these and send a chemical signal. And then they also spend a lot of energy pumping ions across the membrane. So I'm going to talk more about these. And I just want to stress that these are big costs, actually. So our brain, as you're sitting here not getting any exercise, is consuming something like 20% of your energy budget. And about half of that is actually just pumping ions across the membrane. Okay, so how do we understand this mismatch? One possibility is that biology is just not very good at this. Maybe it's just inefficient. Maybe it hasn't been under a lot of selective pressure to get good at this. That's one possibility. I think it's also reasonable to say that maybe this is not really what that cell is doing. So we know that it gets information about the movie, but maybe it's really thinking about something much more interesting. But I'm going to try and argue that it's really the physics of the cellular environment that's leading to these much larger costs. And in particular, information has to be sent over a distance. The components have to be compact, so these computations are carried out by proteins. The information processing has to be timely, And the processing has to occur in the cellular environment, which is warm and watery, and only has a finite or only has specific ways of communication. Okay, so taking a step back, I want to say that biology really uses energy for what I would say are two qualitatively different things. So one is mechanical work done by muscles or macromolecular synthesis. Tasks like this that I would say that thermodynamics really gives a nice accounting for. And as a specific example, a few weeks ago, I went for a hike in the White Mountains with some friends, and I weigh about 100 kilograms. I climbed up about 1,000 meters against the force of gravity, which is something like 10 meters per second squared. And that requires 1 megajoule of work, plus or minus. If you turn this into the food unit, this is the amount of energy that would be turned into heat if I burned a power bar, roughly. So about 250 kilocalories. And you know, I would have had to eat more than 1 power bar for this hike. This was a big hike. And I didn't get this energy back as I went down the hill, right? That was not restorative. But I still want you to think of this as being a rough order of magnitude calculation, right? So this is thermodynamics roughly explains how our cells do mechanical work. But the information processing energy consumption, we have no estimate for how much this should cost. There's no denominator if we want to think about efficiency. There's no satisfactory theory that explains most of these costs. And I want to have a call out for one exception to that, which is that we do have a very nice understanding of why it costs extra energy to proofread molecules to make sure that they really have the correct ordering. So when you replicate DNA that is really reproduced in the correct order, I'm not going to be talking about it in detail, but if you haven't read this paper, in my opinion, this is what the Nobel Prize should have been awarded to Hopfield. It's a beautiful paper, and it explains this quite nicely. Okay, and so what I'm going to be talking about is the cost of processing and communicating information across space. Okay. Okay, so the intracellular communication needs of biological systems are diverse. And I'm going to give 2 examples as sort of motivating examples. So this is a bacteria. This is actually E. coli. And E. coli have a sensor at their nose, which is very interesting. We've done a lot of work thinking about it. But for purposes of this talk, this is taking a measurement of the chemical environment. And then to be useful, it has to communicate that information to the motors sitting over here, something like a micron away. And they need to communicate this information in about 100 milliseconds for it to be useful. At a much larger length scale, this is a neuron, and this is the neuron cell body here. All these red dots that you're seeing here, these are synapses where other neurons are making connections. And these long processes are mostly dendrites. And there, a signal arriving from another neuron needs to be, that information needs to be communicated to the cell body, which might be a millimeter away. And this has to happen in a few milliseconds, so much faster and much longer ranged. To match these diverse needs, the physics that carries out this communication is also quite diverse. So in chemotaxis, this information is carried diffusively. With these sensors, there's enzymes, kinases, that modify a second messenger called Ky. And the message is carried to the motors by diffusion. So phosphorylated Ky diffuses to these motors and tells the motors that they should change their behavior and initiate a tumble. Diffusion would not be fast enough in neurons, and so instead they use electrical signaling. And what happens there is that an ion channel will open, a current will flow in, and this will depolarize the cell body some distance away. I'm going to be talking a lot more about this one. And for now, I want you to note that both of these are very dissipative. So every time that one of these key Y molecules is phosphorylated, that costs an ATP, the energy unit of the cell. And that isn't recovered when the phosphate is removed. It's just lost. And similarly, for this neuronal signaling, these ion channels let currents through. And those currents, you need to pump the ions back out to allow it to signal again. Just to put a number to that, each time one of these ion channels opens, that costs about 10 to the 4 KT. And that's a number that we're going to try and get some understanding of. OK. So to give a bit of an overview of where I'm going, I'm going to start by really going into some detail about electrical communication. And I'm going to tell you how that works mechanistically. And then I'm going to try and derive some energy costs in terms of KT per bit to send information through this electrical communication pathway. Then I'm going to talk about other communication strategies, other physical strategies, and I'm going to compare the cost per bit for those. And then at the end, I'm going to make some initial efforts towards understanding the energy costs of actually running a neuron and putting this into a more realistic geometry. OK, so this is a mechanistic view of the energetics of signaling through electrical currents. So this is a cell membrane in this diagram. A lot of my work is on phase transitions in cell membranes. So it's a little sad to reduce it to just something that separates the intracellular and extracellular space. But that's what it does here. And it also serves as a capacitor. And sitting in this cell membrane, there's the sodium-potassium ATPase. This is the enzyme that uses this 10% or so of your energy budget. And this is a machine that runs through a cycle. And every time it goes through this cycle, it converts chemical energy into separation of ions and charge. So in every cycle, 1 ATP is turned into an ADP plus a phosphate. 3 sodium ions are pumped out of the cell, and 2 potassium ions are pumped into the cell. And the result of this is that the extracellular space is very low potassium, but very high sodium. And the intracellular space has roughly an order of magnitude more potassium and an order of magnitude less sodium. And in addition to this, you note that there's one net charge that leaves the cell during this. And as a result, the inside of the cell is about 70 millivolts lower in potential than the outside. And actually, one thing we can do here already is see that this machine is actually quite efficient. So the energy released by this chemical process is something like 25 KT. And if you take these numbers at face value, they differ from cell to cell. And this one came from Google. But if you take this at face value, there's about 15 KT spent on the entropy of mixing. And maybe you know that a 40th of an EV is room temperature, roughly. So this is about 3 KT of electrical potential energy that's generated. So in net, this uses 25 KT of chemical energy and gets most of that. OK, so why does the sodium-potassium ATPase do this? I would claim that it's not that potassium is better for the cell than sodium. The reason for this is purely so that you can very rapidly send a signal electrically. And the way that works is that there's ion channels, this one I'm going to call the input here, that can mediate currents that are specific to one type of ion or the other. And so, for example, if I open a sodium channel here, because sodium is very high extracellularly and very low intracellularly, there will be a sodium current that rushes in. And if I had a different channel that opened and was specific to potassium ions, then potassium would rush out and there would be a current in the outward direction, right? And so the reason to maintain these gradients is so that you can very rapidly send an electrical signal just by opening a channel that's specific to one of these ions. And the reason that's useful is that you can also have other ion channels or other molecules, though most of them are ion channels, that sense the voltage. So they sense the separation of charge over them. And this is a way that you can very rapidly send a signal, but at the cost of a lot of energy. Okay, so from now on, I'm actually going to be integrating out the sodium-potassium ATPase in many of these details. And in the first part of this talk, I'm going to be considering just the communication part of this, and I'm going to be thinking about a single sender, a single input ion channel, and a single output channel. They're both going to have sizes, sigma I and sigma O for input and output, and they're going to be separated by some distance r. And I'm going to be thinking about how much energy it costs to send signals between them. Okay, so to quantify communication, or one way to quantify communication is through a mutual information rate between some input trajectory and some output trajectory, O of t. And actually, one thing that you're familiar with for a mutual information rate is really your internet connection, right? So when you get information from a website, the rate at which it arrives is the bandwidth or the speed of your internet connection. And you can quantify this. Those are binary variables, but you can think about this for continuous variables also, where the mutual information is really how much the entropy is reduced by an output when you know the input variable. And in some ways, this is going to be a fairly for that, except that we're going to be considering a physical channel, input current, which is going to be inputted by an ion channel here, some output, and for us, what charge density, but localized to some that separates are. And what's different from the usual picture is that we're going to be considering really the thermal fluctuations that are inherent to the medium as causing the noise in this channel. Okay, so in the small signal regime, which these probably are, I can write the information rate as an integral in frequency space that has this form. So this is an integral in frequency space. And at the top of this, there's the input signal power. So this is the power spectrum of the input current fluctuations here, or of the input current. A transmission coefficient. So this is per unit of charge that I let in, how much of it lands over here. So this is really a transmission coefficient, and an output noise spectrum. And I'm going to talk about how we're going to calculate these, but those are the ingredients we need. I can also talk about a dissipation rate. So this is the power that this input channel has to spend to send the signal. And that's going to be given by some generalized input resistance, which could depend on frequency. It won't depend strongly on frequency here. And then again, the input power here. And if integrands by each other, I can get a cost to send a bit that has this form. And here, C of omega is really the energy cost to send a bit of information if I can send it in a thin frequency band close to omega, if that makes sense. Here I haven't made any reference to the physical medium yet, but what I hope you can see is that to calculate this, I need a few things from the physics of the communication channel, right? So I need to be able to calculate this input resistance. I need to calculate the spectrum of thermal fluctuations, and then I need to be able to calculate this transmission coefficient. That's okay. Okay, so I'm going to give a brief overview of how to do this for electrical signaling. The bulk in this sort of minimal model that we're going to write down, we're going to consider to just be a conductor. It has a conductivity of about 1 Siemens per meter. This is a little bit less than the conductivity of seawater. You can think of it as a phenomenological parameter here. And what this means is the current obeys this equation. So there's a current proportional to the gradient and the potential. And there cannot be unscreened charge in the bulk. And that means that the potential in the bulk has to obey Laplace's equation. That's okay. In this model, the membrane is a capacitor with a capacitance of something like 10 to the minus 2 farads per meter squared. And it also supports a charge density. So you can't have unscreened charge in the bulk, but the membrane's a capacitor, so there can be charge separation across it. And we're just going to be thinking about the potential on one side. And so this potential is given by the charge density divided by the capacitance potential in the entire upper half plane, right? Because everywhere it's given by Laplace's equation with those boundary conditions.
But it's simple. Are there free ions? Is it electrolyte?
Electrolytes, yeah. Yeah, so that's why there's no unscreened charge in the bulk. But there can be a charge separation at the membrane.
But there is no field outside. This capacitor.
There can be a field. There's a field in 3 dimensions, but it obeys Laplace's equation.
The charge density on the capacitor is not uniform.
Yeah, that's right. Absolutely. So if it's uniform, then I agree.
It's kind of a fringing field.
Sorry. Absolutely.
If there's a question, either use a microphone or repeat it, just so that people on Zoom can also.
Indeed. So the question is, why is there a field in 3D? And indeed, there wouldn't be if the charge density was uniform. But if there's a spatial variation, then you get a field in 3D. Okay, maybe this is a good segue to this actually. That this means, cool, right? So the dynamics are local in 3 dimensions, but in 2 dimensions you have to invoke this 3D potential. Once you've done that, you can write them this way, and you can write them in a closed form in K-space. This is if you Fourier transform in 2D, but don't Fourier transform the 3rd dimension, you can get a closed form equation. The connoisseurs of such things can see that this isn't local because there's the absolute value of k here, so this is not just a derivative. But this is a closed form equation for the dynamics of the 2-dimensional charge density. And in particular, what this means is that I can calculate a susceptibility. So of these deterministic equations, I can write the charge density as a function of k and omega as a function of the current that I'm applying once I Fourier transform that as well. And I can write the susceptibility in this form, just reading it off from here. Okay, so from this and just a little bit of algebra, really just Fourier transforming from R back, I can do the first thing that we need, right? So we can calculate the susceptibility or the transmission coefficient from input to output. It has this form, and let me just explain a few pieces of this. So the first thing is that it goes like 1 over R. So the longer I want to transmit a signal, the weaker the signal gets with this particular power. It also increases with the square of the output size, which makes sense, right? The larger my receiver is, the more easily I can sense charge, or the charge is proportional to that. And then there's this function u of r over L of omega. This is 1 when r is much smaller than L of omega, and it goes to 0 as they cross. And the way to interpret this, L of omega has this form, and you're used to thinking about an RC time, right? So if I have an RC circuit, there's a time scale associated with that, which is the resistance times the capacitance. Here we have a resistivity and a capacitivity, and when you multiply them together, you get an inverse speed. And this is the speed at which signals propagate along the membrane. And one way to think about why electrical signaling is so fast is that this RC speed is something like 100 meters a second. So signals propagate very quickly on the membrane. The other thing we can get from this is the dissipation kernel, which is really just the resistance here. And it's the resistance from the surface of the input channel to infinity, which is given by 1 over alpha times the radius of the input, as long as this size is much less than the characteristic length scale at that frequency. And so now we can calculate 2 of these pieces, right? The dissipation kernel and also the transmission coefficient. But the thermal spectrum requires more work. And to explain that, this equation that we've derived is really deterministic, right? So this is the deterministic response of the system, but we want the fluctuations, and that just isn't in this, right? And to do that, we're going to use the fluctuation dissipation theorem, which I'm just going to give a very quick overview of. So we're going to use the fluctuation dissipation theorem to get this spectrum of thermal fluctuations. And the essential problem is this, right? This equation, now I've removed the currents, is deterministic and it's dissipative, right? As you follow this, as the fields follow these equations, the energy in them decreases deterministically. And what we really want is the correlation function of charge fluctuations, or alternatively, the structure factor. It's Fourier transform defined this way. And if we had that, we fluctuations at the receiver, right? We're going to use the fluctuation dissipation theorem, but I want to give you a simpler example, which is a force, sorry, an overdamped particle experiencing a force. So if I apply a force to this particle and it's stopped by viscous drag, it'll have a velocity given by mu f, where mu is a coefficient, could be a phenomenological coefficient. And the same forces that cause this drag, these dissipative forces, also have to give thermally driven random motion. And in particular, a different phenomenological constant, this diffusion constant, describes how much this particle is going to randomly move if I let it diffuse for some time tau. And the Einstein relation relates these two. So it says these two phenomenological parameters actually can't be independently determined. They have to have this relationship, that the diffusion constant is equal to the thermal energy times this susceptibility. And it turns out this is the simplest example of a much more general relationship, the fluctuation dissipation theorem, which for these purposes, I can write this way, that the structure factor has to be twice the thermal energy divided by the frequency, but then multiplied by the imaginary part of the susceptibility. So I'm not going to explain more where this comes from, but I think if you think of this as being the equivalent of the Einstein relations, but for a field, that's the right intuition. Okay, and of algebra to get the thermal noise spectrum, and it has this form. And actually, maybe I would just spend a little time talking about the sigma naught to the third. So KT maybe makes sense. The equal time fluctuations should be proportional to sigma naught squared, right? That's the size of this. But because dynamics, there's a characteristic velocity. The length of time or the correlation time of these fluctuations gets another factor of sigma naught. Okay, that will be important in a second. So we have all the pieces of this now, right? And we can put them together to get the energy cost, which has this form. The cost of sending a bit now has this prefactor, but then it scales like the square of the distance between the sender and the receiver, divided by the size of the sender and the size of the receiver, all in units of kBt per bit. And then it has this function which is a function of the separation between them divided by this characteristic length scale. So you can think of this L of omega that comes from this speed of electrical signal propagation as being sort of a viability limit for electrical signaling. So I can send signals as long as they're going less far than that. But if I want to send a signal faster than that, it's going to get very expensive. And the main point that I want you to take from this, I mean, in addition to the specific form, is that this energy cost could be quite large. So if I think about applying this to a cell, R is probably going to be in microns, right? So that's kind of the cellular length scale in neurons, maybe tens of microns even. But proteins are small, maybe a nanometer or two. And so the cost of sending information in this way naturally has this prefactor of microns over nanometers. And the very broad claim that I want to make here is that this is one way to think about the missing factors from the Landauer limit. Okay. So that was electrical signaling, and I want to briefly tell you about a few other signal transduction, a few other ways that signals can be sent. In many cell types, signals are sent diffusively. This can happen in 3 dimensions. So I talked about key Y, right? This is the molecule that lets that signals from the sensor motors by being phosphorylated. In many cell types, calcium is released and diffuses where it binds to things. But there's also membrane-bound signaling molecules. Lipids get phosphorylated, and they also diffuse and bind to receptors. But this is qualitatively different because they're doing so in 2 dimensions. And then at our scale, we use pressure waves, so sound, to communicate. This is very common at the organism scale. And I think it's a bit of a mystery why it doesn't happen at the cellular scale, right? So there are some fringe communities that think that sound is important, but most people, mainstream biology, for the most part, does not think that sound is important for intracellular communication. Okay, and I think what strategy to use when, and maybe as a bit of an amusing communicate with. In this simple picture, an output is just going to be the charge density over one patch of membrane, right? And the first change we're going to make is to imagine that there's many receivers, so many ion channels that are sitting on the membrane, and then they can all receive the signal. And the idea is that we're still going to think about a mutual information between a single scalar input and a single scalar output. But now the output is going to be the sum of the signals on many different channels. That's it. The second change we're going to make is that the real cells are not planar. The calculations so far have been on this sort of infinite sheet. And this is really a model for the soma, right? This cell body here. So we're going to imagine that this is a capacitor that lives on the surface of the sphere at radius r. It's got receivers that are sort of uniformly distributed along the surface of the sphere. And it's going to be shorted at some radius r naught. And you can think about this as being the currents that travel out of these processes, the axons and dendrites, or maybe even the lead current across the membrane. Okay, so I'm just going to very briefly sketch this calculation and try and highlight the things that need to be changed. The transmission coefficient, this now is going to be proportional to the number of channels, and it has this form. The generalized input resistance is unchanged. It's probably slightly different for being on a sphere, but not much. And then the output noise spectrum is the most different. It really has 2 qualitatively different terms, which I'm going to be more explaining. Okay, so so this is the output, and if you imagine looking at the correlation function of this at two different times, there's two terms. One of these comes from the diagonal component, right? So every ion channel has a charge density that's correlated with its own charge density. But there's also an off-diagonal term because different pairs of ion channels or different pairs of receivers have correlations in their charge density at different times. That's okay. They have this form, and importantly, this one is proportional to n, and this one is almost proportional to n squared. Right? Okay. And now, if I'm in the regime where this term dominates, so n is small, then the signal to noise increases linearly as I increase the number of channels, and this sort of makes sense, right? If I increase the number of receivers, I can average their value and I can get a more accurate readout of the input signal, right? But once this term dominates, this information rate will saturate, or the signal to noise will saturate. So the signal squared really, it grows like n squared. And once this term dominates, the noise will also grow as n squared. That happens at a particular value or a particular number of receivers, n star. And n star is the ratio of the size of this inner sphere that shorts out the neuron to the size of an individual receiver. And you can think of this happening maybe around 1,000 channels. And I want to highlight kind of a neat connection to this classic paper by Berg and Purcell. This is a beautiful paper. I highly recommend reading it. But one of the things in this paper, one of the surprising findings of this is that if you want to build a cell that senses the chemical environment, that senses the concentration of ligand or of some chemical that's floating around, you don't need to tile the surface with receptors. And this is because of the structure of diffusion, that if a chemical comes in from infinity, it's very likely to make contact in many different places on the cell surface. And if you really tile the cell surface, the same particle is going to hit it many times and you're going to overcount it. And so you'll just get more noise. You'll get a more, you won't get a less noisy readout. And the scaling is slightly different. It turns out they wouldn't have R not here, they would just have R, so the size of the cell. But I think it's an interesting parallel in the physics that, again, here, you don't need to tile the surface of the cell with voltage sensors to get essentially a perfect readout of the charge density on the cell. Okay, the energy cost also saturates. So once signal to noise saturates, adding more channels does not reduce the cost of sending the energy cost of sending a signal. And in this limit, when the number of receivers is much larger than n star, larger than n star, the cost saturates to this value of R naught over sigma naught. The ratio of the size of this shorting sphere to the size of receptors. And for reasonable values, this could be something of the order of 10 to the 4 kT. In this limit, the cost to send a bit would be something like 10 to the 4 kT. And maybe very tentatively, I would want to claim that maybe this is a functional estimate for why a single channel event should be about this cost. So we can debate whether this is reasonable or not. But this would be the cost of sending a bit to all the other channels, or equivalently to the entire charge density on the surface of the roughly conditions. Okay. So I'm going to start wrapping up. I want to give a few conclusions. So the key points that I want you to take from this, one is that intracellular communication has to be powered to overcome thermal fluctuations. So whether copying a bit really requires energy or not, all of the ways of doing it in the cell do require energy and need to be powered. KBT gives the units to these energetic costs, but they often come with very large key factors that depend on the geometry and the physical mechanism that they're being sent through. Here's the example of electrical signaling. And I've given you a flavor of how you could use calculations like this to try and understand the design principles of specific systems. And in particular, maybe this is the start of an explanation for why ion channels should be this dissipative. And also why it should be possible to have sparse voltage sensors that still basically do a perfect job of reading out the electrical charge on the voltage grid. So these are the main things that I wanted to tell you about, but I guess there's time. I'll just very briefly tell you how this connects to some of the other projects that we're working on. So one of the things that we've been doing a lot of is trying to understand how physical bounds of various sorts constrain biological function. And we've spent a lot of time thinking about E. coli chemosensing. This is part of a very nice collaboration with Thierry to first think about how much information E. coli need to chemotax, so how much information they need to get about their environment to climate gradient, and finding that they're actually very close to the limits of how quickly they can climb given that information. And then follow-ups to try and understand what sets that information. So why couldn't they get more information? And I guess I'd like this to connect in that if we want to try and understand how much energy a specific task should take, we sort of need both sides of this, right? We need to know how much energy it costs to carry out the information processing, and then we also need to quantify what information processing is happening. And then the other side of my work is really trying to understand how critical points and bifurcations could contribute to structure and function. So the last time I gave a physics club, I told you about this work on how bifurcation could integrate information from lots of ion channels. And then another big chunk of my work has been thinking about membrane critical points. And here I think I'd like to work towards an understanding of the energy costs of running these processes, these specific mechanisms.